Hello everyone. In this video, I'm going to quickly explain one of the very important lesson in biology, which is called protein synthesis. So it is the process in which how proteins are made in our body. Now remember, the organelle that make protein is ribosome. Now ribosome cannot do that job by itself without the information come from DNA. So any proteins in our body, let's say the protein that give us the skin color melanin and hair color, skin color gives the melanin, or proteins like muscle proteins or proteins in our enzymes in our body or proteins in the cell membrane or any proteins in our body is they're all made by the ribosome, and ribosome makes that under the command of DNA. So now you know that the DNA is located inside the nucleus. And it's actually located as a large chromosome. The problem here is the ribosome actually located in the cytoplasm. Ribosome. And this ribosome or free in the cytoplasm, or it can be attached with the endoplasmic reticulum. So to make the for ribosomes to make protein, DNA is inside the cell and it cannot come out and tell the ribosome to make a particular protein because DNA is a large molecule. So what DNA do here is DNA select a particular area, whichever proteins it wants to make, at that particular area of the DNA. Let me highlight that part. Give me a second. So let's say DNA wants to make a particular protein and that message is encoded in this section of the DNA. So what DNA does is DNA make a small message, copy of the message from this section. So it makes this copy of that section in a small message called mRNA, or we call messenger RNA. Since this is a small molecule, it can come out. mRNA is right here now. Because it's a small molecule, it can come out. And the mRNA or this message can go to the ribosome. So now this goes to, this is the mRNA. It can go to the ribosome and ribosome reads the message and then it makes the protein according to what the instruction given here. So there are two steps involved in this process. Number one is making mRNA. Step number two is making protein after reading the message which came from the nucleus. So the first step is making mRNA is called transcription. Transcription is the process of making mRNA from DNA. The second step is process of making protein after reading this mRNA message by the ribosome. That is called translation. So there are two steps here. First step is transcription. Transcription. The second step is translation. And these are the two steps in protein synthesis. So now I'm going to quickly explain what is transcription and how it occurs. So the part of the DNA in which this transcription happens, we call this DNA strand. Remember, DNA has two strands, but only one of the strands is going to be used for this, and that strand is called template strand. Template strand. And this template strand, let's say we have the bases here, A, T, C, G, A, C, G, A, T. I just wrote some random bases here. The first step is there's an enzyme called RNA polymerase will come and bind on this section of this DNA which want to be copied. This enzyme is called RNA polymerase. And this RNA polymerase will move along this DNA in this direction. When it moves along, remember it's an enzyme. It can make, its main job is to make RNA. It starts reading this section of the DNA and then start making an mRNA. When it makes the mRNA, remember, it follows the base pairing rule. If the DNA has A, this is mRNA. Remember, RNA do not have thymine, so it corresponds to U. And T here, corresponding base A. C here, corresponding base G. Then G here, C. Then A, U, C, G, G, C, A, U, and T, A. So now we got a new mRNA molecule, and this mRNA may have some mistakes. Cells try to correct it by deleting certain parts and fixing all that. That is called mRNA editing. And finally, the short segment of mRNA will exit the nucleus, and then it goes to the ribosome for doing the second step. So this is the first step, which is making mRNA from DNA. This is called transcription. Now, let's look at the second part. The second part is called translation. So translation happens in the ribosome. So here is the mRNA. So I'm going to use the same mRNA here. UAG, CUG, UAG, CUG, then CUA. So here's the mRNA. And the ribosome start reading the mRNA from one end to the other end, from this side to the other side. So when you look at the ribosome actually has a weird structure. Ribosome actually can read the messages, a large section of RNA actually. So for simple understanding, I'm going to explain that in a short section. So when the ribosome reads the message, it actually reads the message in a set of 3 bases. And the set of 3 bases are called, this is one set, second set, third set. And the set of 3 bases are called codon. So here we have first codon, second codon, third codon. There are 3 codons here. In reality, a ribosomal molecule can read 3 codons at a time. So just for easy understanding, I'm going to draw the ribosome around the 2 codons only, just for our study purpose. So when the ribosome read the codon 1, what happens is, so here's a codon UAG, and remember, the job of the ribosome is to make protein. That is what translation is. So to make the protein, we need amino acids. So somebody has to bring amino acids to the ribosome. That is done by the tRNA. The tRNA has a kind of weird shape. It's like a clover leaf model. And on the top of the tRNA right here, it carries the amino acid. That's the AA stands for amino acid 1. This is the first amino acid. So how do the tRNA bring the amino acid to this position? That is determined by the bases located on the bottom of the tRNA, which correspond to the codon bases here. Here the codon first letter is U, so that base here is going to be A. Here the second letter is A, it's going to be U. And here the G, it's going to be C. So these three bases, A, U, C, are opposite of the codon U, A, G. So we call this three bases are called anticodon. Anti means opposite. Opposite of the codon is anticodon. Now we got the first amino acid. Now the second triplet code is CUG. Now here comes another tRNA molecule, brings another amino acid. How do the ribosome choose that? Because of, again, the base here, C here, so it's going to be G, U here A, and G here, so it's going to be C. That's the anticodon GAC. So now ribosome brought two amino acid. This is the amino acid 2, two of them. The last job what ribosome do is connect these amino acid 1 and amino acid 2. And that bond is called peptide bond. And like this, the ribosome keep moving. Ribosome will keep moving along the mRNA to the right, this direction. And sometimes mRNA has thousands of codon. So as it moves along the mRNA, it will add more amino acids, one after the other, and connect it together and form a chain. Now here we got, including these two, and connect the rest, and we got the polypeptide. It's nothing but protein. So this is how the ribosome reads the message which is given by the mRNA and put the amino acids together one after the other and make protein. And this process is called translation, and this happens in the cytoplasm. And I hope this is helpful to you.